Welcome to the deep dive. Get ready, because today we're going to be exploring something pretty mind-blowing. A complete connectome of an adult fruit fly brain.
Wow.
Yeah, we're talking about mapping every single neuron and synapse, like having a detailed wiring diagram of a thinking machine.
Yeah.
This level of detail has never been achieved before,
Right.
even in simpler organisms. And to help us unpack all of this, we have an expert joining us today.
Happy to be here.
This is going to be fascinating.
It is quite remarkable.
So we'll be diving into not just how this map was created, but also what it's already revealing about how brains function.
Exactly.
To guide us, we've got some incredible research.
Yeah.
A stack of papers hot off the press.
Really cutting edge stuff.
There's a groundbreaking 2018 paper describing how they imaged the whole fly brain at synaptic resolution. Then a 2023 paper on the Flywire project.
Yep.
This massive citizen science effort to proofread and annotate all that data.
Huge amount of data.
And then, of course, we can't forget about
The two 2024 papers.
Yeah.
One analyzing the complete annotated connectome, giving us insights into things like neurotransmitter types and information flow through the brain.
That's amazing.
And the other paper focuses on the fly's visual system.
Okay.
A complete parts list for how a fly sees the world.
Wow.
So by the end of this deep dive, you'll not only grasp the sheer scale of this scientific achievement,
Great.
but also understand what a connectome is and why it's so revolutionary.
Yeah.
And most importantly, you'll walk away with a new appreciation for the complexity and beauty of even a tiny fly brain.
Totally.
Ready to get started?
Let's dive in.
Okay, first things first.
Yeah.
Why the fruit fly?
Right.
What makes this little creature so crucial for brain research?
So the fruit fly, Drosophilum melanogaster, has been a star in biological research for over a century.
Oh, wow.
It's valuable for a couple of reasons.
Okay.
First, it's genetically accessible.
Scientists can manipulate its genes relatively easily to study the effects on development and behavior.
So it's like a tiny buzzing laboratory.
Exactly.
Okay, cool.
And despite its small size, the fruit fly displays surprisingly sophisticated behaviors.
Oh, really?
Like learning, navigation, even courtship, and decision-making.
Huh.
This complexity, combined with its genetic accessibility, makes it a perfect model for understanding the neural basis of behavior.
Okay, so we've got this tiny but mighty fly brain.
Yeah.
Before we get into the mapping, can you give us a quick overview of its structure?
Of course.
Okay.
The adult fly brain consists of about 100,000 neurons.
Okay.
Which is impressive considering it's only about the size of a poppy seed.
Wow.
It's organized into several main parts. The central brain, which handles things like learning and memory. The optic lobes responsible for processing visual information.
Okay.
And the ventral nerve cord, which is analogous to our spinal cord, carrying signals between the brain and the rest of the body.
So even in a tiny brain, there's specialization, different parts for different jobs.
Right.
That makes sense. I guess specialization is a hallmark of complex systems.
Absolutely. Within the brain itself, there are specialized compartments called neuropils.
Okay.
Densely packed regions where synapses, the connections between neurons, occur.
Neuropils, got it.
Yeah.
So we've got this intricate network of neurons and synapses. How on earth do scientists map something so incredibly small and complex?
Right.
This is where it gets really mind-blowing, right?
It is quite a feat.
Yeah.
The key is a technique called volume electron microscopy, or EM for short.
Okay.
It allows us to image the brain at nanometer resolution, which is about 100,000 times smaller than the width of a human hair.
Hold on, nanometer resolution.
Yeah.
That's practically seeing individual molecules.
Yeah.
How do they even slice a brain that thin?
It requires specialized equipment and incredible precision.
Oh.
Imagine slicing a brain thinner than a human hair and taking millions of high-resolution pictures.
Oh.
Then a powerful computer stitches them together to create a 3D models you can explore. It's like having Google Earth, but for a brain.
Okay, so we've got this amazing 3D model of the fly brain.
Yeah.
But how do you go from a bunch of images to an actual connectome?
Right.
A map of all the neural connections.
Yeah.
That seems like another huge leap.
It is, and this is where the incredible Flywire project comes in.
Okay, let's talk Flywire.
Yeah.
From what I've read, this is where the power of the crowd comes in.
It is.
Thousands of volunteers helping to map the brain.
You got it.
So
Flywire is a citizen science project where anyone can contribute to mapping the fly brain connectome.
cool.
Volunteers use specialized software to proofread and refine the computer-generated tracing of neurons in the EM images.
So they're basically making sure the computer got it right, like double-checking its work.
Exactly.
Oh, wow.
It's a bit like connecting the dots, but the dots are synapses and the lines are the intricate branches of neurons.
Fascinating. And just how many people were involved in this massive undertaking?
The Flywire project has had over 100 human annotators contribute over 1,000 edits each.
Wow.
It's a testament to the power of collaborative science, turning a mountain of data into meaningful information.
It's truly awe-inspiring to think about all those people coming together to map a brain, but let's talk numbers. Just how big is this Connectome?
The Flywire project has traced a remarkable 139,255 neurons and identified 54.5 million synapses between them.
Huh.
They've even categorized over 8,400 distinct cell types within the fly brain.
Whoa, 8,400 cell types.
Yeah.
That's way more than anyone expected, right?
It is.
And this is just in one fly brain. Imagine the diversity we'll find when we map the connectomes of other species.
Absolutely.
Okay.
And what's really exciting is that this connectome is incredibly accurate.
Okay.
They compared it with a partially overlapping data set called the Hemmet brain and found very good agreement.
Oh, okay.
So we can be confident that this map is a reliable representation of the fly's neural wiring, which is crucial for drawing meaningful conclusions.
So we've got this incredibly detailed map of the fly brain.
We do.
But what does it actually tell us?
Right.
What secrets are hidden within those connections?
Yeah.
It's not just a pretty picture, right?
It's not.
It's a window into how the fly brain actually works.
Precisely. And one of the first things that surprised researchers was the tight clustering of neurons.
Tight clustering, what does that mean?
Well, previous studies using light microscopy suggested that neurons of the same type were somewhat randomly distributed in certain brain regions.
Okay.
But the EM reconstruction revealed something different.
What's that?
It showed that neurons of the same type actually cluster together much more tightly than we thought.
So it tracks, even in the brain.
Apparently so.
What's the significance of this clustering?
It has many implications.
Like what?
It suggests potential developmental mechanisms. Maybe neurons of the same type are born together and migrate to the same areas.
Huh.
It could also influence how neurons sample information from their surroundings.
Okay.
If neurons of the same type are clustered together, they might be processing similar types of input.
Interesting. So it's not just about the connections themselves, but also about the spatial arrangement of those connections.
That's right.
What else have we learned from this connectome?
One of the most exciting discoveries has been the ability to predict neurotransmitter types.
Wait, you can tell what kind of chemical signal a neuron uses just by looking at its structure in the EM images?
We can.
How is that possible?
It's thanks to artificial intelligence.
Oh, wow.
Researchers trained a neural network to analyze the EM images and predict the neurotransmitters used by each neuron based on subtle structural features of the synapses.
That's incredible. So knowing which neurons use which chemicals to communicate is a game changer.
It is.
It's like understanding the language of the brain.
Exactly.
And with AI, we can now predict this for an entire connectome. This opens up incredible possibilities for studying how information flows and how behaviors are generated.
Absolutely. This AI-based prediction is a huge GE step forward.
Yeah.
Previously, obtaining this kind of information required laborious and time-consuming experimental methods.
Right.
Now we have a tool that can rapidly analyze the entire connectome and give us a good idea of the chemical signaling landscape in the brain.
So by combining the connectome with these neurotransmitter predictions, we're getting a much richer understanding of how information flows through the brain, right?
You got it. The concept of information flow in the brain is crucial.
Okay.
Think of it like a relay race. A signal passes from one neuron to the next, each one processing and transforming the message along the way.
Okay.
By mapping the connections, we can trace the entire race course and understand how the brain transforms input into output.
This is truly revolutionary. It's like going from a static map of roads to actually seeing the traffic flow.
It is.
But we've only scratched the surface of what we can learn from this connectome, right?
Absolutely. This is just the beginning.
Yeah.
There are so many more questions to explore.
So
now that we have a complete wiring diagram, we can start to build computational models that simulate the activity of the fly brain.
So like creating a virtual fly brain in a computer.
Exactly.
Could we manipulate the connections, change the activity of certain neurons, and see how it affects the fly's behavior in the simulation?
Exactly. We could play what-if games with the brain to figure out what makes it tick.
Wow.
We can test hypotheses that would be impossible to test in the real world.
This is amazing. It feels like we're on the verge of a whole new era in brain research.
It does feel that way.
But can we really extrapolate from flies to humans?
Right.
Our brains are vastly more complex.
They are.
What are the limitations of this approach?
That's true. Obviously, flies don't have the same cognitive abilities as humans.
Right.
They don't have language consciousness or the capacity for abstract thought.
So there are certain aspects of human intelligence that we simply can't study in flies.
That's right. But that doesn't mean we can't learn anything valuable.
Okay.
Even studying simpler forms of intelligence can give us insights into the neural mechanisms underlying more complex forms.
Huh.
At the level of individual neurons and synapses, the basic principles of neural communication are remarkably conserved across species.
So the language of the brain is, to some extent, universal.
Yes. And while the human brain has billions of neurons compared to the fly's 100,000,
Right.
there are fundamental organizational principles that we share.
Okay.
For example, both fly and human brains are organized into specialized regions with distinct functions, and both use a variety of neurotransmitters to communicate between neurons.
So by studying the fly brain, we can learn about the basic building blocks of intelligence.
Exactly. It's like starting with a simpler model system to understand the fundamental principles before tackling the more challenging cases.
Right.
What we learn from flies can inform our research on more complex brains.
Fascinating. Speaking of seeing the world,
Yeah?
let's dive into the fly's visual system. I'm particularly interested in this because it's like getting a glimpse into a completely different way of experiencing reality.
It is.
Yeah.
The fly's visual system is truly remarkable.
Yeah.
And thanks to this Connectome, we now have a complete parts list for how it works.
Wow.
The 2024 paper focusing on the optic lobe provides an unprecedented level of detail. They identified over 700 cell types in the optic lobe alone.
Wow.
That's many more than we previously knew existed.
700 cell types just for vision.
Oh,
That's mind-blowing.
It is.
What kind of diversity are we talking about?
These cell types vary widely in size, shape, and connectivity.
Okay.
Some are tiny and process information from a very small region of the visual field. Others are much larger and integrate information from a wider area.
Right.
This reflects the complex computations involved in vision, extracting information about motion color objects, depth. It's all happening in these intricate circuits of the optic lobe.
So each cell type is like a specialized worker contributing to the overall task of seeing. I'm curious, what can we infer about the function of these different cell types based on their connections?
That's one of the most exciting aspects of this Connectome.
Really?
By analyzing the wiring diagram, researchers can start to make educated guesses about the roles of different cell types.
Okay.
For example, some cells are connected in ways that suggest they're involved in detecting motion. Others seem to be specialized for recognizing objects.
Right.
Still others might be involved in processing color information.
So it's like having a map of the brain, A and D, a key that tells you what each part is likely doing.
Exactly.
Amazing. Is there anything else that stands out about the organization of the fly's visual system?
One interesting finding is that many cell types are arranged to cover the entire visual field in a relatively uniform way.
Okay.
This is called tiling, and it ensures efficient processing of visual information.
Okay.
It's like having a grid of sensors,
Yeah.
each responsible for a small patch of the visual world.
So the fly's eye is divided into these tiny processing units, each with its own specialized team of neurons.
Exactly.
It's incredibly efficient.
It is.
We've got this incredibly detailed map of the fly's visual system.
We do.
But how does the fly actually perceive the world?
Right.
What does its visual experience look like?
That's a question that's fascinated scientists for decades.
Yeah.
And while this Connectome doesn't give us a direct answer, it provides invaluable clues.
Okay.
We can now start to build models that simulate the activity of these visual circuits and see how they respond to different visual stimuli. We can explore how the fly detects motion, recognizes objects, and navigates its environment.
It's like we're starting to see the world through the fly's eyes, and who knows what amazing insights we'll uncover.
Right.
This is just the beginning.
The beginning of a new era?
I'm already buzzing with excitement for what the future holds for Connectome research.
Me too.
But for now, let's take a moment to appreciate the sheer wonder of this scientific feat.
I agree.
We've mapped an entire brain down to the level of individual synapses.
It's incredible.
It's a testament to human ingenuity and the power of collaborative science.
Absolutely.
Well said. We've covered a lot of ground in this deep dive.
Yeah.
But there's still one more exciting aspect of this research to explore.
All right.
The fly's extraordinary oscillar circuit.
Wow.
It's a specialized visual system that plays a crucial role in flight control and navigation.
That sounds fascinating.
Stay tuned as we delve into the fascinating world of the fly's third eye.
I can't wait. Welcome back to our deep dive into the fly brain connectome.
Yeah, we've covered some truly amazing ground already, from the basics of the fruit fly brain to the technology and human effort behind this connectome.
Right, but now it's time to zoom out a bit and tackle some broader questions.
Okay, I'm ready for the big picture stuff.
What are the implications of this research for our understanding of intelligence? Can we really learn about the human brain from studying flies? These are the questions we'll be exploring.
Those are some big, fascinating questions.
Yeah.
Where do we even begin?
Well, let's start with one of the most fundamental questions in neuroscience.
Okay.
How do brains produce behavior? How do these intricate networks of neurons and synapses give rise to the complex actions, thoughts, and feelings we experience?
It's kind of like the mind-body problem,
Yeah.
but at the level of neurons and circuits.
Exactly. And for centuries, this question has remained a mystery. But with connectomes, we finally have a tool that could help us unlock it.
So by mapping the connections, we can start to understand the computations that underlie behavior.
Think of it this way. If you want to understand how a computer works, you need to know more than just the individual components.
Right.
You need to know how those components are connected, how they interact with each other.
Okay.
The connectome gives us that wiring diagram for the brain.
Hold on. Is it really fair to compare a fly brain to a computer? I mean, they're both processing information,
Yeah.
but are they doing it in the same way?
That's a great point. It's important to remember that brains are far more complex than computers. Even a tiny fly brain with its millions of connections.
Right.
But that's where the power of computational modeling comes in.
Okay.
We can use computers to simulate the activity of those millions of connections and see how they give rise to behavior.
So we can create virtual fly brains in a computer and then experiment with them.
Exactly. We can manipulate the connections, change the strength of synapses, add or remove neurons, and see how it affects the fly's behavior in the simulation.
So it's like a virtual laboratory for brain research.
Yeah.
We can test hypotheses that would be impossible to test in the real world. That's incredible.
It is a powerful tool, and this is where the true potential of connectomes lies.
Okay.
They provide a framework for understanding the relationship between brain structure and function.
This is all incredibly exciting, but I can't help but wonder, can we really apply what we learn from flies to humans?
Right.
I mean, our brains are so much more complex.
That's a valid concern.
Yeah, of course.
There are limitations to this approach.
Like what?
Flies don't have the same cognitive abilities as humans.
Okay.
They don't have language consciousness or the capacity for abstract thought. But there are also surprising similarities between fly and human brains.
Okay.
At the level of individual neurons and synapses, the basic principles of neural communication are remarkably conserved across species.
So even though our brains are vastly different in scale and complexity, there's a shared language, a fundamental set of rules.
Exactly. And while the human brain has billions of neurons compared to the fly's 100,000, there are fundamental organizational principles that we share.
Okay.
Both fly and human brains are organized into specialized regions, and both use a variety of neurotransmitters to communicate between neurons.
So by studying the fly brain, we can learn about the basic building blocks of intelligence, the foundation upon which more complex brains are built.
Precisely. It's about understanding the spectrum of intelligence from simple to complex.
Okay.
By studying the diversity of brains across species, we can gain a deeper appreciation for the evolution of intelligence.
Okay, that makes sense. So even though flies may not be pondering existential questions or writing poetry, their brains can still teach us a lot about the fundamental principles of how brains work.
Exactly. And what we learn from flies can inform our research on more complex brains.
Yeah.
It's like starting with a simpler model system to understand the fundamental principles before tackling the more challenging cases.
Right. This has been a really thought-provoking conversation.
Good.
It's amazing to think that a tiny fly brain can teach us so much about the nature of intelligence.
It is remarkable.
But there's one more area I'd like to touch on.
Okay.
We've been talking about the potential benefits of this research, but are there also potential risks?
That's an important question. As with any powerful technology, there are always potential downsides that need to be carefully considered.
So what are some of the potential risks or ethical considerations associated with connectome research?
Well, one concern is the potential for misuse of this technology. For example, if we gain the ability to manipulate neural circuits with precision, We need to be extremely careful about how that power is used.
Right. If we could potentially alter someone's thoughts, emotions, or behaviors by tweaking their neural connections, that raises some serious ethical questions.
Absolutely. It's essential that we engage in open and honest discussions about the ethical implications of this research as it advances.
Okay.
We need to establish clear guidelines and regulations to ensure that this technology is used responsibly and for the benefit of humanity.
It sounds like we need to be having those conversations now before the technology outpaces our ethical understanding.
I agree. Open dialogue and careful consideration are crucial. But despite these challenges, the potential benefits of Connectome research are vast.
Okay.
Imagine a future where we can diagnose and treat brain disorders with unprecedented precision, where we can understand and address the root causes of conditions like Alzheimer's, Parkinson's, and autism.
That's an incredible vision and a powerful reminder of why this research is so important.
It is.
But we need to proceed with caution and wisdom. Okay, I think we've covered a lot of ground here, from the nitty-gritty details of connectome mapping to the grand implications for understanding intelligence and the ethical considerations we need to address.
We have, and I'm glad we could explore those broader aspects, but we still have one fascinating piece of the puzzle to uncover.
Oh.
The fly's third eye, the ocellular circuit.
Right. We can't forget about that it's a specialized visual system that plays a crucial role in flight control and navigation. Stay tuned as we delve into the fascinating world of the fly's ocellular circuit. Welcome back to the deep dive. We've explored the fly's intricate visual system, delved into the broader implications of connectome research, and now it's time for a truly unique part of the fly's sensory world.
Right.
The ocellular circuit.
Yeah, it's a fascinating area of study, often overlooked,
Mm.
but crucial to the fly's ability to navigate and survive.
Okay, so remind me what exactly is a ocellular circuit. It's like a third eye, right?
You could say that flies have those two large compound eyes we typically picture,
Right.
but they also have three simple eyes called ocelli located on the top of their head.
3 eyes. Okay, that eye is wild. What do these ocelli actually do? Are they like backup eyes in case the compound eyes malfunction?
Not quite. The ocelli have the very specific function. They're primarily involved in detecting changes in light intensity,
Okay.
which helps the fly maintain stable flight and navigate its environment.
Hmm.
Think of it like a built-in horizon detector.
So they're more like specialized light sensors,
Yeah,
providing information about the fly's orientation.
exactly. And what's remarkable is that this seemingly simple function is carried out by a surprisingly complex neural circuit.
Really?
Thanks to the Flywire Connectome, we now have a detailed map of this ocellular circuit,
Okay.
allowing us to trace the connections from the photoreceptors in the ocelli all the way to the motor neurons that control the fly's wings and muscles.
So we can actually see how light information is transformed into action. That's incredible. Can you walk us through this circuit?
Sure.
What are the key players?
The main pathway from the ocelli to the central brain is through a group of neurons called OCG01 interneurons.
Okay.
These neurons receive input from the ocelli and relay that information to other parts of the brain, including regions involved in motor control.
So the OCG01 neurons are like the middlemen between the ocelli and the muscles, translating light information into instructions for movement.
A perfect analogy. And here's where it gets even more interesting.
Okay.
There are two types of OCG01 neurons.
Oh.
Some are predicted to be excitatory, meaning they activate the neurons they connect to.
Okay.
Others are predicted to be inhibitory, meaning they suppress the activity of their target neurons.
So it's a push-pull system. Some neurons telling the muscles to contract, others telling them to relax.
Precisely. This balance of excitation and inhibition is crucial for fine-tuning the fly's movements, ensuring they're smooth and precise.
So that makes sense. But how does the fly integrate information from the ocelli with information from its compound eyes?
Right.
Are these two visual systems working completely independently?
That's a great question.
Yeah.
And the answer is no, they're not independent.
Okay.
The ocellular circuit is actually tightly integrated with the visual pathways from the compound eyes.
Oh, wow.
This allows the fly to combine information about light intensity from the ocelli with information about motion, color, and objects from the compound eyes,
Great.
creating a richer and more comprehensive representation of the world.
So it's a multisensory experience. The fly's brain is blending information from all its senses to create a more complete picture.
Exactly. This integration of sensory information is essential for the fly's survival, allowing it to navigate its environment, effectively avoid obstacles, and find food.
Wow.
It's a beautiful example of how different parts of the brain work together seamlessly to guide behavior.
This has been an incredible journey. We've gone from the microscopic world of synapses and neurons to the grand questions of intelligence, consciousness, and the ethics of brain research. It's truly mind-boggling to think that so much complexity can be packed into such a tiny brain.
I agree. The fly-brain connectome is a testament to the power of scientific inquiry and the endless wonders of the natural world.
We've explored the intricate wiring of the fly brain, uncovered hidden patterns in its neural architecture, and considered the broader implications of this groundbreaking research for understanding intelligence and the potential for future medical breakthroughs. We even got a glimpse into the world through the fly's eyes, including its remarkable third eye, the ocular circuit.
It's truly a remarkable achievement, this mapping of an entire brain.
Yeah.
And it's just the beginning.
Right.
As we continue to map the connectomes of more complex brains, who knows what incredible discoveries await us.
It's an exciting time to be exploring the brain, that's for sure. Thank you so much for joining us on this deep dive into the fly-brain connectome. Until next time, keep exploring, keep questioning, and never stop wondering.